So 100%. I'm going to discuss medical cannabis grade quality considerations for international offtake. And in this regard, I always like to start with the end in mind. I think if you understand medical grade cannabis, or at least inflorescence or flower, it's a good way to start working your way backwards in terms of understanding the dietary supplement market or the CBD wellness market, and then going back to ultimately what would be the recreational market. So basically, in this discussion, I want to really define what is meant by medical grade cannabis or GMP flower. If someone refers to that term, what do they mean when they insinuate that? And then a quick touch on GACP, GMP, the pharmaceutical need for harmonization, PICs and ICH, and then also look at COAs, because that's sometimes the best way to actually understand what is meant by the quality, and a touch on ISO17025 that relates to lab testing. And then at the end, I'd like to really go into what international offtake is currently looking for with regards to medical cannabis, and then countries which are open to international offtake, the importance of stability data, as that's often not on a COA, but it ultimately affects the shelf life and stability of the final product. And probably more in line with GMP, because the previous may be more related to GACP, is a discussion briefly on whether to follow CFR 111 or 210-211. So let's get started. And I would say today's conversation for me is really inspired by cannabis inflorescence for medical purposes, where the US Pharmacopoeia recently put out a paper on the quality considerations, and also guidance was provided by the FDA on cannabis and cannabis-derived compounds, the quality considerations for clinical research. I like to always use an example that works at the moment for medical cannabis, which in this instance would be Bedrocan. Bedrocan is well known throughout anyone in the European flower or inflorescence markets. They call it floss on their end, but essentially there's 5 varietals of products intended for sale into the pharmaceutical market. Here we can see specifications around the percentage of delta-9 or tetrahydrocannabinol, as well as any additional cannabinoids, the dosage form in flowers. It's got a batch number, lot numbers, etc. The date it was harvested, if it was irradiated, what was the dose. This is a recent COA, so it relates to September. Who was the packager? Who was the lab for testing? What was followed in terms of the testing method? Here it is the Dutch monograph. If you do want a copy of the Dutch monograph, I'm happy to email it through to you, or if I'm possible, I can perhaps add it as a PDF in the attached discussion on Zoom. So just to give you an idea, this is for flower that's ultimately going to be checked and released. So if this was flower that was prepared by Bedrocan and sold into Germany, they would go through this monograph, and the key things to look at is the appearance, the identity of the product, looking at the microscopy, TLC in terms of that foreign material, fineness. We can perhaps discuss this in a little more detail, but generally the requirements are outlined within the monograph, where there's specifications and a need to conform to those specifications. In terms of the micro testing, this will relate more to European pharmacopoeia, specifically 514 for aerobic microbial counts, as well as yeast and mold counts, as well as gram-negative bacteria. So this gives you an idea of what they're using in terms of following which standard, the colony-forming units that need to be not exceeded. And then the important one here is aflatoxins, specifically outlining which chapter within the European Pharmacopoeia is being used as a base point, and then pesticides. I would say, however, the Dutch have identified that their pesticide testing list is somewhat reduced. I think it does exclude some GCMS workflow tests, where it's stipulated actually here on the second page what isn't covered by their pesticide testing. I know that our previous speaker at Maya also referred to the presence of metals, so specifically which European Pharmacopoeia chapter to follow regards to that. Here it's actually a monograph specification, where I believe their method is ICP-OES, detection limits, and then loss of drying under 2232, and then any assays and additional testing via the monograph. So I like to use this as an example, because when we talk about the monograph, or specifically these COAs, it perhaps gives an indication to individuals what exactly is being tested and why. Bedrocan is a good model to look at as an example because it applies almost exclusively to the entire Dutch framework, and it also has supplied to Germany and parts of Western Europe for a while. And then in terms of Europe, a lot of the import has been from Canada, where they would typically follow a USP method or methods in terms of evaluation. And if anyone's been tracking that space, they'll be quite familiar with the conversion that Canada has been doing from GPP, good production practices, to GMP. For me, I'm going to step back, and there's quite a bit of a debate here because what is medical grade cannabis? Cannabis is cannabis. It really comes down to the need for consistency. The whole point of this extensive battery of testing is to establish consistency of the product. And if you're going to enter into clinical trials for an investigational new drug, consistency is really the overriding concern, especially if we're using flower or the actual inflorescence for entry into a clinical trial. Stability of that genetics over a period of years is what Bedrocan claims to have achieved. That's debatable and a discussion point. But whenever you're dealing with a herbal or anything derived from a herbal base, it's a little different to a traditional API, which the room would know. And the need for consistency and controlling the genetics and the cultivation conditions to allow for that consistent product is critical. And if you're going to get clinical evidence, you're going to need to eventually take this to a new drug application and to further marketing authorizations. And that's why the need for consistency is really what this all aims towards achieving when we use a word like GMP flower. For me personally, I like extraction workflows. So sometimes easier to manage the product when it's gone through an extraction workflow because you're able to really narrow into specifics. That said, the one benefit of flowers that anyone will know is the entourage effect due to the terpenes and the combination of the cannabinoids. But that said, we need to get a consistent product. And that's really the aim that is being put forward by companies like Bedrocan, is trying to get a consistent flower to market that can be used for pharmaceutical use. The other point to discuss is the need for consistency on testing. ISO17025 obviously sets the outline for testing standards. Increasingly, you want to have consistency in terms of the testing and the standards. There's a USP reference here in terms of your internal references within testing are also critical because you need to make sure that you're not using old standards and anyone who's been testing flower or extracts currently in the market, even between labs, the differences in the testing is really what can be quite frustrating. And I'd also say it's very frustrating if you get a test result locally and then you export your product and it gets received and you get a completely different test result. That need for harmonization is really critical around the testing standards. And ultimately, all of this is geared towards establishing safety, efficacy, and quality of the product, but at a cost, which is really what the distinction is in terms of price points when we talk about medical cannabis. Now, the following slide is more of an internal document that I personally use. So please take it with a pinch of salt. But what I've tried to do with this document is I've outlined the different types of tests that usually relate to inflorescence, as well as different methodologies in terms of the instrumentation, and try to provide USP guidance as well as related EU guidance to that test method. It is in no way comprehensive. There's definitely going to be additional chapters that relate to this, but I try to use it as an example just to start giving more information out to the public domain, so that, you know, the more information we have, the more we can start to get a clearer understanding. I'll use specifically potency as an example. Potency can be tested a couple ways: TLC, high performance TLC, NIR, FTIR, GC. I would say the workhorse and the accepted method would generally relate to HPLC. There also be proponents of an LC-MS workflow. However, here, what you're limited by is the matrix and the excessive need for dilutions. So I would say if we want to use HPLC as the method here to define potency, here's some good guidance in terms of validating that methodology, which USP chapter, if you're using that specific form of the pharmacopoeia, or which European pharmacopoeia guidance could you use or look at. And I'd say this might help in terms of starting to outline, you know, what are the costs that are going to be involved? If I'm going to talk about in-house analytics, and in GMP, we talk a lot about critical process parameters. If I'm going to be doing any form of consistent testing in-house, my in-house analytics, what's critical to me and what's not? Can I outsource the pesticide testing, which is usually more expensive, to a third-party test lab? And what additional concerns may be there ultimately? I would say stability is something to be looked at. It's not on a COA. It's definitely done post process, but it can be trended for. Residual solvents is something that you wouldn't relate to specifically in fluorescence or flower. It would definitely be more in terms of if you've gone for an extraction workflow. Interesting topics like extractables related to packaging. Here's some guidance on that, but that's also becoming an interesting topic. I'd say the main one you should look at as a first point of entry is articles of botanical origins. And there's definitely more chapters that can relate to this space, but I just wanted to use this as a good reference point to just get the conversation going in terms of testing. I wouldn't be doing it any justice if I didn't bring in harmonization around this. I'm not going to spend too much time on this because the chapter is clearly defined, but the key things is to get reproducibility, accuracy, repeatability, a good level of specificity, limits of detections and quantification need to be defined, along with linearity and range. But I'll leave that to an expert in the field that really can address the validation around the testing methodologies. What I want to jump into is just a discussion on the need for harmonization and standardization of testing globally. Everyone likes to, I like to use monopoly as an example. You want to succeed in this business. You want to get it right, pass beginning and collect your $200. You do not want to have a situation where we test locally and then we send it off and there's a disparity between results between labs. Obviously, ISO17025 covers this or avoids this if everyone's of the same standard, but it's also important to discuss the need for consensus on methodology. If someone's using a GC for potency locally and then you have sent it off to a lab that's using an HPLC, those are things to start looking out for. The need that the internal standards, if they're being used in methods and the validation of those methods, that they exist, they're in place, and that those standards actually have good stability, because the last thing you want to do is have your entire testing compared to a standard that's old or exceeded shelf life. On harmonization, the big one for me is pesticides. Very often when I deal with international offtake, they'll use the Canadian list as an example of the pesticides they need tested for, which to me makes little sense if you're sending a product from South Africa. So whether you're using the Californian, the Oregon, or the Canadian pesticide lists, almost feels like there needs to be a broader discussion as it relates to what testing for pesticides is required from which regions of the world. I'd like to jump forward and be respectful of time. The topic of GACP is a big one to discuss because it actually forms the basis of the quality of the product. Establishing whether that should be indoor or outdoor and what are the risk mitigation factors of each approach is almost a discussion to in itself. I would say for me, the big thing around GMP is what I'd like to close out with, which is the discussion around the different sub-chapters, so 111, 210, 211. PICs, obviously, the nice thing here is everyone should know in the room about PICs, but I'll just touch on it briefly. It's essentially the Pharmaceutical Inspection Conventional Cooperation Scheme, where I believe 54 countries are members, including South Africa, being the only country in Africa that's member of PICs. But generally, the objective being the harmonization of GMP across the regions, so that we could essentially say that a product manufactured under GMP in South Africa meets a similar GMP standard across Australia or North America if they're a member of PICs. And then obviously the ICH is the definitions there, but really helps drive harmonization between pharmacopoeia. So countries that are open to offtake. I always like to advise anyone who's looking at offtake, firstly target a country with an existing medical program. The reason being that there's domestic need for that medicinal grade cannabis. So you're almost assured of consumption. Also ensure that there's no protectionist policies in place. I think it's been established now for a while that Canada has enacted a form of protectionism around imports. I've included the reference from MJ Biz Daily, where specifically products that are being medical grade products that are being brought into Canada are getting stopped at Port Authority, where this example is as recent as the 6th of August of this year. Ideally, there will be a point in the future where there would be a mutually recognized agreement or MRA for cannabis in Europe, making it a lot easier to move products in and out of different regions of Europe. However, that might be a bit optimistic until there's changes within the WHO. Clarity on COAs. This for me, if you're in the space where you're engaging with offtake, make sure you have a complete understanding of the testing that will be required once you ship and for it to be cleared and received on the opposite end. So if you're targeting Europe, understanding what it is required in terms of their COAs and their standards. There's a big push for stability data. So the need that you start putting products into stability testing is becoming more and more prevalent within the offtake community. I would also say pay close attention to where there is the best offtake currently happening. Israel overtook Germany as the main importer of medical grade cannabis in July of this year. But this was also followed up in September, not too recently, where they've also increased the stringency around their medical import specifications. So it was a very nice place to send product to in terms of offtake. But, as countries become more and more informed about medical grade cannabis, including extracts related to medical grade cannabis, there's going to be a need for closer scrutiny regarding the GSEP, the licensing, the extent to which the regulators are enforcing that, as well as GMP standards. So stability, essentially, if you need guidance here, you can have a look at ICH topic Q1A revision 2. Stability testing, obviously, of new drugs and finished pharmaceutical products. I would just say the purpose of stability testing is really to provide evidence on how the quality of a drug substance varies under time, exposed to temperature, humidity, and light. So in a nutshell, you basically place your product into stability with a COA at the point of introduction. And then when you pull your sample, let's say 6 months later under aggravated or accelerated stability, or a year later, you get an indication of how's that product degraded. This is relevant because I'll use the Dutch. They accept a tolerance of 20% from labeling claim, whereas the Germans will only accept 10% variation from labeling claim. And anyone who's really interested in this, just be aware, obviously, of the different climatic zones. So where you're shipping your product to, you want to develop stability data specifically to the region you're planning to ship to. So South Africa, we would be in zone 2. So if you're sending to Australia, or you're sending off to Italy, or to specifically some other country, you need to just make sure that your stability data supports that. If you go for a more extreme form of stability, just be aware that obviously with a product like flour, terpenes don't hold up well. So sometimes a more extreme version of stability might do an injustice towards the results from that product. And then I'd say my last slide, really, I'd like to touch on 21 CFR 111 related to dietary supplements and how that compares obviously with Part 210 and 211 related to pharmaceuticals. I have seen that 99% of the US manufacturing space falls under 111 for dietary supplements. And, this is quite important because very often what I see is an active is produced, so a specific extracted product, being whether that's isolate or distillate, is actually supplied under 111 and then done under final packaging under 211 and then sold via that route, or just sent via 111. And I think this is important because I would anticipate that the grade of pretty much all isolates that are even coming onto shelves in the country that are being imported are being derived from obviously 111 and packaged or co-packaged or contract manufactured under 211 if it is even that stringent. But I would also like to maybe bring in a discussion on this point, because generally when we manufacture for export, very often the traditional view would be that we need to target 210, 211. However, if our products are coming in being basically extracted under dietary supplements guideline for isolates and being basically formulated perhaps here under 211. It's just a topic that could be quite an interesting one to discuss, but I'm going to be respectful of time and look to wrap up here for our next speaker, as I see we're approaching 3 o'clock. The only thing I would say is I've done some chats on extraction previously, so I'm happy to share information and videos on that. And if you need to get hold of me, feel free to reach out to me or to David. I'm sure you'll be happy to pass on my details, but I'm quite passionate about the space, and I'm really passionate about South Africa realizing the potential to be a player in the medical cannabis scheme and also in the CBD wellness space. So for me, it's a golden opportunity, and I enjoy events like this where we get to collaborate, come together, and share viewpoints and expertise from different areas. And I'm always open to learning more and engaging. So absolutely, feel free to reach out to me, because there's a lot that I'm still learning. I think there's no expert in cannabis or medical cannabis globally, really. Everyone is in a learning curve, and it's just a fascinating field. So thank you so much for the time and the opportunity to present.